under this Act. New varieties as section 15(1) of the Act. Extant varieties, section 2J15(2), notified under the Seed Act 1966. Farmer varieties and variety of common knowledge, essentially derived varieties. Dust tests to be conducted at the multi-location minimum two crop season, and shall be a official description of the variety. Special test when dust test fail to establish distinctiveness. Trees and vines have the on-site test. Protection period in this Act. Protection period for the trees and vines is 18 years, but initially it is 9 years and extended another 9 years after deposition of this fee of extension period. Other than trees and vines, its protection period is 15 years, and it is firstly 6 years, and repeatedly after the deposition of another fee is 9 years. Extant variety is totally 15 years. As per the Seed Act, the notified the varieties under Seed Acts are the extant varieties are registered without any dust test. Plant variety registration. The variety finalized the dust test guidelines for the registration of now 180 crop varieties, crop species covering cereals, pulses, millets, oilseed species, vegetables, flowers, medicinal and aromatic plants. And up to December 2022, 5,112 certificates were issued. And across the country, there are many dust test centers for the crops. Dust test for the candidate variety conducted at crop specific center, and the data is submitted to the authority for further analysis. Authority has established National Seed Bank and the Field Gene Bank. National Gene Bank is required for the conservation of the variety protected for that period, that protection period, because if there is any dispute in this protection period, then the court can ask the sample from the National Gene Bank, that is the required for the protection period. And in the Field Gene Bank, the true plants were planted there for that protection period. It is also for this as a sample of the protected variety. Plant Variety Journal of India and maintains the National Register of Plant Varieties as headquarters and branch offices. The authority maintains publish this Plant Variety Journal monthly on the first working day of the month, and maintains the National Register of Plant Varieties. And the details of the varieties registered are entered in the National Register of Plant Varieties. Now I am coming on this status of horticultural crops in the varieties of the horticultural crops received in the cucurbits are 544. Details are here in the table, you can see, and the registered registration certificate issued in this in this group of the crops. You can see the details of the horticultural crops registration, and these details are here in the table. The status of horticultural crops of registration. This is the dust test guidelines of different crop species, and this is the 180 crop species open for registration. And in this group, the cereals, legumes are other than the horticultural crops, but this flowers, ornamental crops, fruits, medicinal aromatic plants are here listed in the list. The guidelines under the notification, these guidelines are Dolgus bean, carrot varieties as gold, snake gold, and ivy gold, just notified in the Plant Variety Journal, and yet to be notified by the ministry. And other guidelines are under the development process: broccoli, chow chow, sponge gourd, lettuce, vegetable kale, brown top millet, and no gold. Brown top millet is millet. Thank you. If anyone has any query.
Kindly keep your question here. It will be asked in the all presentations. The time you will start. So we are in hurry. Thank you, Doctor. Okay, thank you. Next speaker, Dr. R.M. Sharma, who is a medical scientist at IARI, who is going to speak on the walnut in India, status, constraint, and prospect. Dr. Sharma, please.
Okay. Honorable Chairman, Co-Chairman, reporters, former directors of CISH, my colleague, Chief Organizer, Dwidiji, faculty members, friends and students. Today I am going to here deliver a small discussion on Indian walnut industry. That is a very important crop for all aspects. So I will discuss only one. We are having total 535 accessions, and out of these 91% are being reserved at Central Institute of Temperate Horticulture. Why walnut? Because you know that our temperate industry is based on difficult terrains, except Kashmir Valley. So soils are very poor, soil depth is very poor, they are rain fed. So we being a very hardy crop and having long shelf life, because many times transport facilities are not available. So if you grow peach, plum, and apricot and cherry, so they are highly perishable. So this is one of the advantage with this crop. Highly nutritious. I will discuss in the next slide. Hardy nature, it is highly drought hardy. High domestic demand. It has been found that in last 15 years, 30% domestic demand of walnut has been increased because with the growing awareness of the health and all these things. And even overseas demand is also there. Huge employment generation, because nowadays majority of the walnut is being exported in terms of kernel. So kernel is being extracted at domestic level. So more than 2 lakhs people in Kashmir Valley are only engaged with walnut kernel extraction. So this is a huge industry. Good source of foreign exchange, around 74-75 crore rupees worth we export the walnut. Precious wood being used for different purposes, furniture, musical instrument, sports, kitchen goods, gun stocks, and all these things. Recently, a trial has been conducted in 2021, and it has been found that male catkins of walnut are having very high health promoting activity. So its powder in bread making can be mixed up to 2.5% in the wheat flour to develop a value added bread. Cell biochar as the electrode, because generally the walnut cell is not being utilized. But many exporters in Jammu region, which are having a huge infrastructure, they crush the walnut shell powder, and it is being used in bulk in civil aviation and railway ministry, because it is used for cleaning of the heavy machinery. And the electrode is also being used from the biochar of the this walnut shell, which is being used for detection the water pollution with cadmium, lead, mercury, and copper. So this is thus the nutritive value, and you find this is the comparative table of the different temperate nuts. And walnut is having very high fat content, especially polyunsaturated fatty acids and alpha-lipoic acid and arginine. This is a very interesting study which was conducted in 2021. That this study was conducted on the undergraduate student in South Australia in a university, that how semester system create a mental stress on the student. So they have created 2 groups. One group was given 56 gram walnut, and for 16 or 17 weeks this study was carried out, and it was found it is very very important nut to alleviate the stress in the undergraduate student. These are the findings of that study. Basically, it protects against the negative effect of academic stress and metabolic biomarkers. Walnut consumption improves sleep. Academic stress was associated with lower gut molecule diversity, which is being decreased under the pressure of academic activities. So this is our current status of the world in India. So total walnut production of the world is around 2,314,000 tons, and we contribute only 36,000 walnut, which is hardly 1.55% of the total walnut production of the world. This is our country's condition, and 91% walnut is being produced only in Jammu and Kashmir. Now it is coming in very big in Arunachal Pradesh also. So this is our per capita walnut consumption. We are only 20 gram per person per year as compared to other countries. This is our walnut export figure, because 10 or 15 years back, the whole walnut was exported in terms of shell. But nowadays, only kernel is being exported. 95% of our export in the terms of kernel. And these are our top 5 destinations for export, and we export about 2482 metric ton walnut worth of 73.98, around 74 crores. And our 72.82% export goes to UAE, France, UK, Djibouti, and Germany. Now, simultaneously, because I told you that our total production is 36,000 ton, our domestic demand is 60,000 tons. So we are not in the position to fill our own demand. That is why we are importing a huge amount of walnut from different countries. This is very interesting that one variety is there, chandra, which has been developed by US, but its planting material has been imported by Chile, and now Chile is producing better quality chandra walnut as compared to US. So our maximum import of walnut is from Chile. Around 70% our import is from Chile, and total we spent around 474 crore rupees to import the walnut to cater our domestic demand. Traders also. Now these are the constraints of our walnut industry. First of all, non-availability of organizing procedure. I will discuss one by one. We don't have ideal varieties. Elevation constraint, the variety which has been developed for 50 meter above mean sea level, that cannot be grown in on 100 meter above mean sea level. Color will be affected. Non-availability of quality planting material is the most serious problem. Low kernel yield, low nut, because most of our varieties are terminal data. So our yield hardly 35 or 40 kg per tree. Poor huge investment for export. I told you that we invest around 474 crore rupees to import the walnut. Poor technical know-how for kernel extraction, because the kernel is being extracted at domestic level. So many times they soak the shelled walnut, and if the water is not pure or water is not clean, ultimately there are the chances of the infection of the E. coli or salmonella. Stick restriction under J&K Prevention Act. I will have separate slide. Non-availability of our root stock, biotic stress, no systematic improvement. Genetic erosion, that is a very important part, because we are concentrating only good kernel plant. Otherwise, rest of the plants are going to die. So we have to conserve those plants also. And constraints for trader, they are having different story. So first point is non-availability of organized growth orchard, because walnut is by and large propagated by grafting. So a large size of signwood is required. And on one tree, you will not get more than 20 or 30 signwood from one tree. So because of non-availability, we don't have sufficient quantity of signwood. Non-availability of ideal variety, because walnut variety must have 3-4 characteristics. One, it should be late blooming, early harvesting, good kernel quality, and late fruiting. These 4 characteristics are there, but we don't have any variety which is having 100% lateral bearing. This is the photograph of Turkey. This is the lateral bearing walnut. We don't have such kind of variety. They are having yield per tree, but we are having between 30 to 40 kg per tree. So because we don't have such kind of varieties, we have some introduced varieties. These are some prominent varieties of Jammu and Kashmir and Himachal. These are few varieties we have developed through the clonal selections. So Hamdan, Suleiman, these 2 varieties from Srinagar, rest are from Solan. Now these 10 hybrids have been selected. 10 selections have been developed by Central Institute of Temperate Horticulture. They are having around maximum 54% kernel recovery. And this is one variety. It has been highest kernel recovery which has been developed, around 61.4% kernel. So this variety has been developed by Sri Kashmir Agricultural University and has been released. And it is an excellent variety. And export rating of this variety is 9.5 out of 10. Elevation constant, I already told you that if you are going to develop a variety for 1500 meter above mean sea level, you cannot grow it on 100 meter above mean sea level. Because of high temperature and intense light, the kernel shell will be hard and kernel color will also be kept. This data I have collected through the personal communication. This is the serious problem in walnut industry because we don't have sufficient plant. I discussed with the HOD of Dr. Bayes from my university, Sudan. They produce around 3,000 plants, having demand more than 10,000 plants. And plant cost 250 rupees per plant. Iswas Kashmir producing 7,000 to 8,000, having 20,000 demand. State is having demand of 2 lakhs plants, but only able to propagate 6,000 to 7,000 plants only. And this is the nursery man, which it was trained by these people when we were working there. So he alone producing 6,000 plants from very small structure, but the plant cost is very high. In Uttarakhand, Mr. Sudhir Chadda has also tried to sell the plants of standard variety, but the cost of plant was 800 rupees. And this is the forest department collaboration with SIT. I will show the separate slide for that. This is that farmer which is going to establish on nursery and producing around 6,000 plants of walnut. He is having 60 to 70% success. Now, walnut area expansion in Uttarakhand because Central Institute of Temperate Horticulture and Forest Department of Uttarakhand, they have made they have signed a MOU, and under this project, because this project under JICA, Japan International Cooperative Agency, so around 35,000 plants have been supplied by Central Institute of Temperate Horticulture to the Uttarakhand for raising the income of the farmers. And they have also started to develop their own plants. Around they are also producing 3,000 to 4,000 plants per annum. And this is the statement of the director said that we have covered on around 200 hectare land in Uttarakhand by supplying the 35,000 plants of water. Poor technical know-how of Kannada, I told you that if you are not using clean water, ultimately the chances of the salmonella and E. coli will be there and the product will be unfit for the export. This is a very strict law, JNK prevention of specified tree, because some trees in Jammu and Kashmir are having special status like salix, walnut, cedrus, and pine. You cannot cut from even your own property. So you have to seek the permission from the divisional commissioner, and which is almost impossible. So in 2021, Horticulture Department have written to the government that kindly lift this ban because we are not in the position to plant fresh trees. So in December 2022, Government of India has asked to the Jammu and Kashmir government, give me the list of the law which you want to repeal after revocation of the Article 370. So this law has been listed in that list. Hopefully, in the coming year, this law will be dissolved. These are some objective research root stock because one generally hardy crops are difficult to propagate by tissue culture. So some root stock of walnut have been developed like BX211 and RX1. These are easy to propagate by tissue culture. That is the beauty of these two root stocks. And this is root stock which has been developed in 2022. These are some major biotech effects of temperate region in walnut. First one is walnut weevil. Generally, walnut fruit crop is not there in walnut. But if walnut weevil infestation there, you will not get even a single fruit. Heavy infestation is there, and it can be controlled at the time of flowering. Otherwise, you cannot control it. Anthracnose is a serious problem. This is a very uncommon problem in walnut because this is the parasite. We call it mistletoe, and scientific name is Biscum alum. So its berries are very tasty. So when birds eat them, and walnut trees are huge, so they sit on top and parasite through excreta, and after some years, the parasite starts to grow on the walnut tree, and within 3 or 4 years, whole walnut tree will die. These are some ideal characteristics of a so kernel is being exported, so you must have extract color quality. So these are some certain constraint of the traders. Mixture of seedling produce, they don't have organized market, high domestic demand, no material for the market, internet supply of the produce, land is there on which walnut can be transplanted. High domestic demand is there. Its nutritive property are very high. So keeping in view, the walnut industry needs to be strengthened in our country. Thank you.
Very good.
Thank you.
So now we can talk to next speaker without slide.
I'm going to present my topic on the bioresources of Northeast India, scientific validation, commercial utilization, and conservation strategies. You know that Northeast India consists of 8 different states, which is very rich in that very beautiful state, rich in their that cultural identity, rich in its traditional and ethnicity, and which is very rich in that biodiversity. You know that it is one of the center of the 36 mega biodiversity hotspots. In addition to the other plant resources, this region is very rich in horticultural resources. This region exists different climates, like in Arunachal Pradesh, very temperate climate. In Mizoram, it is subtropical, and in Assam, in some parts, it is just like the tropical. Because of its variation in that climatic conditions, there is existing that a good number of tropical, temperate, and that subtropical fruits, vegetables in these regions. And among that, in the biodiversity of horticultural crops in this region, considerable diversity exists in their morphology, their physiological characteristics, adaptability, as well as in the distributions. And most, in addition to the commercial varieties of horticultural crops, this region exists a big number of underutilized horticultural crops. If we see that habitat diversity of horticultural crops in the northeastern region, basically they are in that still in many states like that Mizoram, there are primary forests are there, and many that horticultural crops are there in the primary forest. In addition to that, they are in that abandoned land, home gardens, and agroforestry systems. In each and every household of Northeast India, there are that at the backyards of their home, it exists that home gardens, and where they used to have either one or the other types of that fruit crops. Yes, among the different fruit crops, the most important one is that citrus. It is believed that the citrus original home is that in the Northeast India. Even in the one Jin sanctuary is there, that is in the Nokrek Biosphere Reserve, which is located at that West Khasi Hills of that Meghalaya, that West Garo Hills, sorry. That in that region is still that there are the primitive citrus species are there. So in the northeastern region, major citrus species are citron, that lemon, mandarin, pomelo, sweet orange, satkara, rough lemon. These are some of the commercial and the semi-wild citrus species grown in this particular region. In this area, there are 23 species, 53 varieties, and 7 probable hybrids. Among them, the most important citrus species that Khasi mandarin, which is grown commercially, and because of the presence of one particular hill, that is Khasi Hill, which is located in that Meghalaya, the species is named as that Khasi mandarin, and which is very popular because of its sweet and that sour blending, that equal blend of this sweet and sour. In addition to that, other species like Assam lemon, that Medica, then rough lemon, and other types of lemons are there in this particular region. And there are the 23 hybrids have been reported from this particular region in this citrus. Among them, the most important one is that Khasi mandarin, as I have mentioned. And recently, last year, that Meghalaya has got that GI tag for this Khasi mandarin, which is one of the very good type of among the 4 different types of mandarin varieties grown in India. This is one of the highly demanded mandarins in in our country. So recently it got the GI tag. And in addition to the khasi mandarin, other citrus species are rough lemon. That there are different types of rough lemon grown in this particular region. And some are that highly acidic and some are that sweet in their taste. And they are even named as that by the that local names that in the which are there in the different parts of the region. In addition to that, another important citrus species is that Assam lemon. And this is the only that citrus species that which is found that throughout the year. Even that it produces flowers throughout the year and it produces fruits also throughout the year. But during winter time, because of that temperature and other factors, that production it used to be that get reduced as compared to the summer time. Lime, another important species found in some that states of this northeastern region. Two varieties are basically found: Abhayapuri lime and that Karimganj lime, which are there that the names are given only because of the name where it is available. Another important citrus species that Citrus macroptera, which is commonly known as that Hotkara or Satkara in that different regions, that different states of northeastern India. And in northeast India, this species is found in that Meghalaya, Manipur, Nagaland, and Mizoram, not in other states. And but because of its high medicinal values, now that people from Bangladesh and Myanmar, they used to come and they used to that collect these fruits at a very cheaper rate. So that and these fruits have been found to be that have that very good that bioactive compounds as well as very good high amount of this antioxidants. Another important species where that albedo portion is edible, and albedo or the spongy white portions, which is that very thick in this particular species, which is known as Citrus medica. And this species is also that having that good source of that medicine. And different local cultivars are there in this northeastern region. Another particular species that Ilasi nimbu, and this species is very famous for its that the flavor just like that of the that cardamom. That's why it is known as that Ilasi nimbu. Then all the that citrus species have been found to that have their that different ethnomedicinal properties, and the local people they have been used this citrus species either in one form or the other form for different types of that medicine. So this is also that another that Meghalaya recently got. I mentioned that the GI tag for two citrus species. Among in addition to that citrus mandarin orange, this is that another one which is Citrus indica, and it is believed to be that the primitive citrus species and which is found only in Meghalaya and Arunachal Pradesh in that wild in the jungles of Arunachal Pradesh and Meghalaya, which is known as that Memong Narang. And in addition to that, some other citrus species are also found that wild and semi-wild stage. Citrus letipas, then this is Adazamir, that which is commonly, which is also having that flavor just like that of that ginger. That's why it is known as that Adazamir, and scientifically it is Citrus assamensis. So Isanggensis, another that which is locally known as Isang pepeda, and this is this recently that the researchers found that this particular species is a very good rootstock, which is having that cold hardiness properties. Then Megalogycarpa, locally known that commonly known as that sour pomelo, which is fruits are similar to that of the pomelo but very sour in taste. Then in addition to that another that all those species that Kasai lemon, and that since last 3-4 years this particular fruit is found only in that Manipur. One particular village of Manipur, which is known as that Kasai village, which is located in that Ukrul district of Manipur. This species is found, and very recently, Manipur government that have started that promotion, that observing that Kasai Lemon Festival every year in the month of January, February to promote that Kasai Lemon that producers of that particular village. And Manipur got that GI tag for this particular variety. In addition to that, those citrus, another important species that banana. In banana, in addition to that normal that our common that commercial varieties, that one very important variety is that Bhimkol, which is seeded banana, but it is that recently actually that some private companies have started producing some baby food from this banana. It is belongs to that Balbiciana group. And in addition to that, other that commercial bananas. A number of that wild bananas are there, like Musha ornata, which is basically that ornamental banana. Then Anset glaucum, which is actually that just related genera. It belongs to that related genera of this Musha, that is Anset. So these are some of the wild bananas of northeastern region. So pineapples, basically that Q and Queen are that commercial pineapple cultivars of that region. But in addition to that, some other local varieties like Jaldap and Lakhat, these are also found in this particular region. And in jackfruit, in addition to that normal that Artocarpus heterophyllus, in these regions that Artocarpus lacosa or that monkey jack is also that found that growing in that wild that forest of northeastern region. And in mango, we have that some wild cultivars like that wild variety that types of mango like Mangifera sylvatica, which is found in that Assam as well as that Tripura. So in addition to that other commercial that varieties, a number of that underutilized fruits are found in this northeastern region. As I mentioned in my previous slides, that the one of the major source of that of this horticultural crop is that backyard or that bari system. Each and every that household of northeastern regions, they have there in the just backside of their house, they have that they used to cultivate either one or the other types of these underutilized fruits. And among that underutilized fruits, some are that very promising and have different that medicinal properties as well as that some other that very high nutritive value. So these are some of the underutilized that fruits in this northeastern region. I'm mentioning only very few, but in addition to that, there are lots of underutilized fruits which needs that good promotion. And the Dillenia is that one of the that which is commonly known as that elephant apple, one of the that very popular that wild fruits of that Assam. Then Phyllanthus acidus, or which is known as that star amla. Then Garcinia pedunculata. In Garcinia, actually, that 3-4 different species are found in this northeast India. This pedunculata is very common in that Assam and Manipur. Then that lancifolia, which is very common in that Mizoram. In different parts of Mizoram, this lancifolia species is there. Then another species that Garcinia, that xanthochymus, that is also found that very that grown without any commercial cultivations in different parts of this northeastern region. So most of them have been that all that wild fruits which I have mentioned in my previous slide. All of them, they have that the medicinal uses in northeastern regions, in the tribal states like that Meghalaya, Mizoram, Manipur. In each and every villages, some that traditional healers are there, and they used to use this kind of that wild edible fruits or that to heal that different that problems or different diseases that which are faced by the general that people of the rural people. Yes, although I have mentioned a very good number of that commercial as well as that non-commercial that horticultural resources in this particular region, but still that region that there are lots of gaps in that plant genetic resources conservation. So first of all that I'd like to mention about the traditional that practices which have been not yet been that documented, and it has been transmitted from one generation to that another generations so that only orally. So there is need to be that documentation of that plant that traditional that practices which are followed by the village people. In addition to that, the government, there is gaps in that policy plan and the implementations of that programs. The government has not even taken that proper steps for conservation of the horticultural resources. Number 3, that there is lack of that research in the genetic diversity. Very few research have been carried out to that for about that genetic diversity of these horticultural resources in this particular region. Even most of them, most of the wild edible fruits in this particular region have not yet been documented. Yes. So now I'd like to discuss about that conservation strategy. So first of all, that collection and evaluation of the germplasm. Yes, it is that prime important to conserve the horticultural resources. We have to that the indigenous resources should be collected. They have to be documented and they have to be evaluated and characterized for the desired horticultural traits. Number 2, that rootstock improvement programs. Although there are different rootstocks found in this region, for different fruit crops, but there have not been that proper research work have been carried out about that rootstock improvement program. Then production of superior quality fruits is also that another important program to be carried out in this particular region. Then development of feasible and economically viable techniques for quality improvement, that is also that most needed for this particular region. Then development of viable processes for extraction of that color pigment, flavonoids, and etc., especially that from that processing waste of different horticultural crops. Then that evaluation of the non-edible fruits for production of the citric acid. As I have mentioned that about the citrus, although a number of non-edible citrus species are there, but those are actually that not used by that people, and they are getting waste when that season comes. So it is the high time to that evaluate that non-edible fruits, because there may be that good scope for production of the citric acid as well as the pectin. And production of the flavonoids can also be carried out from the different fruits which have not been taken in this particular locations. So future R&D strategies, thorough assessment of the indigenous genetic resources, and their employment in the strategic breeding programs involving the national and exotic varieties is of prime importance. Then assessment of the nutritive values of this particular fruits, and including the measurement of the ORAC values to judge that antioxidant capacity. Yes, I am sure that all of them definitely will have the different important things. Then conversion of the homestead garden to the nutrition. I'm just about to finish. Nutrition garden, the homestead gardens to the nutrition gardens. It is the high time to evaluate, to characterize them, to know their nutritional qualities, and to cultivate the highly nutritional fruits and vegetables in the backyard of the houses. So at last, I'd like to just say that appropriate national policy, action, and plan and programs are very much important to conserve these valuable resources and their sustainable utilization. Number two, intensive inventory and documentation of the useful species is of highly required to conserve this highly valuable horticultural resources in this particular region. And some pilot programs should be immediately initiated for plantation, domestication, as well as cultivation of these species, as well as for the conservation of these highly demanding species. Thank you.
Now I will call upon the next speaker, Dr. K. Gupta, who is going to speak on the genetic improvement in onion for wider adaptability.
Respected chairman, co-chairman, dignitaries, and all the participating scientists. Good afternoon to all of you. I am going to present a talk on genetic improvement in onion for wider adaptability. As you are know that onion is an important cash crop and grown and consumed throughout the world. It is basically a full season crop, mainly grown in Rabi season, but it can be cultivated during Kharif and late Kharif. The diverse genetic material is required for improvement and need of the changing demand. ICR DOGR is working as a nodal agency for onion and garlic research and development, and also working as a national active germplasm site for the collection and evaluation of onion and garlic germplasm. We have conducted exploration in collaboration with NBPGR and covered 16 states and collected number of onion and garlic germplasm which have dark red, light red, white, and yellow onion germplasm. The yield of available varieties are low as compared to the world average. It is 16.4 ton per hectare, which is very less in comparison to the world average. So, for increasing the productivity to meet our world average demand, there is need to research on yielding variety with good storability and desirable horticultural traits and availability throughout the year. Now, plant genetic resources in onion. At DOGR, Directorate of Onion and Garlic Research, 1429 germplasm has been collected and being maintained, including dark red, light red, white, yellow, rose type, multiplier type, and also wild types. In addition to that, we have also collected number of accessions from abroad, including USA, Taiwan, Germany, with the help of NBPGR. We have also taken IC number from NBPGR and seed of these lines has been submitted for long-term storage. So we have characterized all the collected germplasm for the desirable traits. So this is the list of germplasm being maintained at the DOGR. This slide show the variability available in the germplasm like dark red to white, yellow type, multiplier type, rose type also. This slide show also variability available in the wild germplasm because wild germplasm has some specific feature which can transfer in the cultivated lines. Now, genetic improvement in onion. Based on the available germplasm, we have developed 10 onion varieties which are released and notified at national level. Among these, Bhima Super, Bhima Red, Bhima Dark Red, Bhima Supra and Sweta suitable for Kharif season, whereas Bhima Raj, Bhima Shakti, Bhima Kiran, Bhima Light Red and Bhima Sweta suitable for the Rabi season. Out of these, Bhima Shakti, Bhima Super, Bhima Red, Bhima Raj and Bhima Subra also grown during the late Kharif, which are the very important season for the availability of onion. We have also registered these variety with the PPVFRS for the protection. These all the 10 onion variety are in demand and adopted by farmers in all over the country. In addition to that, 10 onion variety released by DOGR, about 72 onion varieties released in India by different public organizations, including DOGR varieties. And our 10 DOGR varieties have several features and suitable for the Kharif, late Kharif as well as Rabi season. I want to mention here the main crop is Rabi season and 50 to 60% produce available from the Rabi season and only 20 to 25% from Kharif and 20 to 25% from late Kharif season. So if Kharif crop is failed, then the price fluctuation is started. So there is a need to develop a variety for year-round availability. So price fluctuation can be minimized. These are our this slide show photographs of our variety. In addition to that, we have characterized the germplasm, and here some of the germplasm have found is specific feature for specific traits which are mentioned here. And in addition to that, we have also started breeding program, and out of 70 two building lines. We have identified some of the lines for the earliness, high yielding, free from double and bolters, bulb shape and size, storability and seasonal availability, and out of which at present 12 elite lines are being evaluated in network trial, All India Network Research on Onion and Garlic Research. In addition to that, we have registered 4 genetic stock with NBPGR for the specific traits like DOGR 1203 for early maturing, DOGR 1549 for early as well as suitable for Kharif and Rabi season, DOGR 1666 for waterlogging tolerance, DOGR 1656 for drought tolerance, and DOGR 1168 for bolting, and DOGR 1523 for early maturing and suitable for Kharif and Rabi with 5 to 6 cloves in case of multiplier only. We are also working on bolting tolerance. Bolting is a major problem during Kharif season, late Kharif season. If bolting occurs in seed crop, it is desirable, but bolting occurs during the main crop, it is undesirable. So we have identified some lines which have less bolting, less than 5%, but have a high good bulb yield, which are under the trial and entered in the network trial for the further multi-location evaluation. We are working on multiplier onion and our 5 multiplier onion under the network trial, out of which 1 multiplier onion found suitable for the South India, that is Maharashtra and Gujarat zone 5. We are also evaluated for green onion for green as a green utilization. Generally, bulb is used for the consumption purpose, but immature tops are also used as a green vegetable. So we have identified 3 multiplier onions, so which aggregatum type, which are suitable for the green use. Now, in addition to that open pollinated variety, we have also working on hybrid F1 hybrid and developed several hybrids with using 5 male style lines, and now presently you have 6 F1 lines under multi-location evaluation trial through the network. We have also developed 1 early maturing line which has registered with the NBPGR as a genetic stock, which are used for development of early lines. So 18 crosses were made, or now in 5th generation 4 populations were found early in maturity with high yield and have 100% net fall, uniform net fall. Net fall is a desirable character in case of onion, which indicate the maturity. We are also working on the integration breeding, long day and short day onion, by the crossing between short day and long day onion. At Srinagar, we have started this long back 2018-19, and presently we have 6 population in desirable direction. These are also under multiplication for the multi-location evaluation through the network trial. Another technology, this is the SEX technology. We have screened our 32 available varieties, including the DOGR, and found only 3 varieties are suitable for the SEX. SEX is the technique which are used for raising the early Kharif crop, because early Kharif crop is very economic during the Kharif season, because it produced during the October and price fluctuation coming in the September, October. So this technique has been recommended by the network, our network system, ECRIP system. For this technology, Bhima Super, Bhima Dark Red, and Bhima Sweta are suitable for the producing the sets. Sets are used instead of seedlings for the production of commercial bulb. We have also working on wild onion and evaluated for the flowering behavior and Allium tuberosum and Allium actum flowering time is January to May, whereas fistulosum flowering March and Esculanicum flowering time is February. All the variety has been registered with the PPFRA and other variety also from the country which are registered with the PPFRA listed here for the information, because protection of plant variety with PPFRA today is the prerequisite for the protection of their variety and availability of genuine seed with the farmers. So we have developed 10 onion variety which are suitable. Some are suitable for Kharif season, some are for Rabi season, and another for late Kharif. But there is a need to develop more and more variety which are suitable for year-round availability. Thank you.
Thank you. Now I will call upon Dr. He is working at National at Pune. He is going to speak on the towards molecular breeding and identification of genomic region for various traits and development of alien specific markers for various traits. Another try to listen.
Okay. Respected Chairman, Co-Chairman, Rapporteurs and distinguished delegates. I'm going to present some part of my work towards molecular breeding in grapes, which involves identification of genomic regions for berry traits and development of allele-specific markers for berry traits. So, just as a brief introduction, I would like to say that grape in India is grown over an area of 140,000 with a production of almost 3,200,000 metric ton annually. And the majority of the grape is used for table purpose. And for table purpose, for its commercial importance, we require loose bunches, bold berries, uniform berry size. These are the desirable traits for good marketability of the table grape. So at our institute, there is focus on the development of varieties with loose bunch and bold berries. But through conventional breeding, development of variety takes long time, or simply because grape is a perennial crop, which it has a very long gestation time. It is heterozygous, highly heterozygous in fact. We face inbreeding depression, linkage drag is there, and of course selection and testing of progenesis requires very long period. So it is very prudent to identify the genomic regions which control these important traits. And so that information can be used for the development of markers for early selection of the progeny, as well as for the identification of candidate genes, major candidate genes, which can be used for variety development through biotechnology process. So in this study, we use two approaches because generally for identification of genomic features, we need a large segregating population, but being a perennial crop, many times it is difficult. But the genome-wide association analysis provides alternate approach where we can use randomly selected germplasm in our germplasm. We have almost 475 accessions in our germplasm. So we selected randomly selected grape genotype. And fortunately, for last 15 years, we have some structured breeding programs, and we have developed two very good segregating populations. In being a heterozygous crop in grapes, we consider F1 hybrid itself a segregating population. So we have two F1 segregating population in our institute. So one of that population we use for CTM. And since we were using F1 segregating population for developing linkage maps, we had to use sugar test. Of course, now softwares are available where we can do this kind of analysis. So we use those software. For genotyping, we used next generation sequencing-based genotyping by sequencing, which helps us to identify the single nucleotide polymorphic markers. And in this part, we did genotyping data for 3 years for 12 bunch and berry traits. So this is is just a broad outline of the association mapping. We take randomly selected genotypes, phenotype them, and genotype them. Then we do a market rate correlation analysis using different approaches, and then identify closely associated markers with the trait, and then we can further explore to identify the genes in that particular region. And of course, this is just a broad aspect. So we selected 128 grave genotypes, which were randomly selected using computer-based software. There was no bias in the selection of those genotypes. And based on ES phenotyping data on these traits, we can see that we have a wide range of those traits in our selected genotypes. But when we look at the distribution pattern of these traits, only in case of very length and very width, we could see the normal distribution. So, and this is the distribution pattern for these traits. Here we get typical bell curve for these two traits, whereas for many of the traits we got a distortion. So that is the thing. And then for a marker analysis, we use genotyping by sequencing analysis. And since we have their genome available in the public domain, that was used as reference genome for the identification of SNPs. And of course, we could identify large number of SNPs across all samples, almost more than 50 lakh SNPs for identification. But we get lots of missing data, and then minor alleles had to be removed, and poor quality data were removed, and finally we retained only about 69,000 SNPs for further analysis. Using this SNP data, we did principal component analysis to know whether there is some structure is there in these genotypes, because grape in our earlier studies, we had found that in a collection very narrow genetic diversity. There are many of the genotypes are interrelated, and if we don't consider that in our analysis, there will be some false positive analysis. So this analysis was very important, and we could see that there was some structure, like some of the genotypes were tightly grouped together. So this information we had to use for our association analysis. So this particular approach, generally the model of course doesn't uses this population structure. That's why we could identify large number of marker trait associations, and several of the SNPs were pleiotropic. That means they were so one SNP was found to be associated with more than one trait. So this is some data about that. But when we use population structure into consideration during our analysis, the number of these associations were reduced because there the possibility of false positive was limited, and we got maximum number of associations for derivatives, and for other traits also we could get 1 or 2 marks. So this was about association mapping. When we went for detailed linkage mapping, we had a segregating population of random black rose into complex sequence. There again, this about 100 habits we used for SNP detection using GBS, and we have a software join map which utilizes strategy for linkage mapping. We developed linkage map for that, and the map data and phenotyping data was used for PTL mapping for this. So this is the basic data which we obtained from our GBS analysis, and those SNP markers, because both the parents are heterozygous, so we classify them into 3 classes. One class was the SNPs which were heterozygous in CBR, and those SNPs were used for developing linkage map of CBR. There was another category of SNPs which were heterozygous in CS, and the data was used for Thompson sequence. And then these 2 classes, other SNPs were there which were heterozygous in both German black rose as well as CS. So those SNPs were used for development of integrated maps. So we developed 3 different maps from this data. So this is the, I think, clear picture is not there. So this is the linkage map of CBR. There were 1,716 SNPs located on this linkage map, and average distance was 1.29 centimorgan. So we can say it was a very dense linkage map. So similarly for Thompson series also we got 1.11 centimorgan average distance. Again, very distant. Of course, we were having some gaps were there, in those regions we couldn't find this thing, but overall the density was 5. And this, in fact, in literature also, all the maps which are available in different genotypes, same kind of density. I'll take another one. And this is again phenotypic data of severe interface. However, those hybrids are having wide variability for the selective. So, and in fact, in this population, we could get normal distribution for many traits, as we can see the value of Shapiro fever. And this is the distribution pattern. In QTL mapping, now map data, we had map data for our parents. We have a phenotyping data of population. So map data and phenotyping data we subjected to interval mapping, and we could identify some QTL regions for different traits. And here is this thing. In fact, there are many more QTLs, but I'm presenting here only the QTL regions which was showing LOD value of 5 or more than 5. So these are very significant. And there is analysis which tells us how much LOD should be considered. So for grape, it was coming to 5.5. So even about 5.5, there were a few GPS which are quite having quite high LOD. And these explain large proportion of the addition for this. So these are important. Now, after identifying these as SNPs, we need to validate them further. So for that, we developed allele-specific primers. So suppose this is a SNP CA. So we developed one primer forward and another. This thing for each SNP, we developed 2 forward primers, each one having one weight. And to differentiate on simple agarose-based analysis, we added 15 extra bases in one of the primers. So 15 difference can be easily differentiated on agarose. So I think there is a photograph which is not shown. One photograph is there. One of the allele-specific marker I wanted to show, which is segregating clearly between low berry rate and high berry rate. Somehow this photograph is not shown here. And then we try to identify the candidate genes also in those regions, and these genes were identified. And we try to do estimation of genomic breeding values, and we could see that a set of FNP markers could were able to predict these states with quite good prediction activity. So this data will be used for genomic selection of design. This 4,000 SNPs we have shortlisted, which gives good prediction activity. So this is just to conclude that we could develop densely case maps for our parents, both the parents as well as integrated maps, which were used for QTL identification, and same maps can be used for identification of QTLs for other traits. And then association mapping also, we could identify good number of SNP markers which were associated with traits, and these SNPs and QTLs will be useful for the identification of candidate genes as well as development of markers for their use in molecules. So this work was funded by ESA, and many students helped us to gather the data. And these are the varieties which we have developed during last year. This is our margin indica. They are with medicinal properties. This is our margin naveen, which is a very good flavored variety for table purpose. This is Marjorie Shama, a good pulp, crunchy filling. And then this is Marjorie Kishmish for reti purpose. So these are the 4 varieties our institute has developed very soon. So thank you very much.
Very nice presented. Now I would like to call upon Dr. D. Pandey from the Lucknow. Dr. Pandey is going to speak on genetic diversity of fauna and grape. A role for climate resilient agriculture.
कैसे सही Respected chairman, co-chairman, and dear reporters and dear participants, I am going to present the अरे ये वाला है वो कोई सा खोलिए वो तो एक ही होगा. Genetic diversity of amla and bale, a boon for climate resilient horticulture. और त्रिवेदी जी जरा देखिए इसमें कैसे होगा, हमको ज्यादा आइडिया नहीं है. ये वाले बटन
Climate change, as you know, it is creating a lot of problems. Particularly, the human activity has caused around 1.1 degree centigrade of warming today, which impacted agriculture, biodiversity, and human society. And most of the in agriculture, land degradation is one of the major problems, which is due to drought, flood, rising sea, tropical storms, disturbed food availability, reduced access of to food and food quality. And you see the most of the part of the globe is is affected by the climate change, and in India particularly the hilly area are mostly affected as compared to the southern part. And to mitigate this impact, climate change directly or indirectly causes negative impact on quality attributes and nutritional quality of fresh food and might increase severe production problems. Changing land use for development purpose leading to the farming household with marginal land to cultivate. In India, around 176 million hectare area is lying as a wasteland, which is in future we are going to cultivate particularly these food crops in wasteland because the good land we are not going to get because of the population pressure. So specific food crops suited to such land can be feasible and rapid alternative to mitigate climate change impact and provide continuous nutritional supply. The bale and avla are such an important crop which can be grown in these type of the soil, and they are the crops which can tolerate the climate aberration in a good way, and you can get good yield during the extreme of the temperatures or the low of the temperatures. So horticulture crops, especially fruit, nut species, and plantation crops, have potential to withstand harsh climatic conditions. Jamun and guava exhibited tolerance to salinity but susceptible to frost and drought. Bale and avla are the most sturdy and grow in very harsh environment. Bale, which is native to India, primary center of origin, has mythological significance which dates back to Vedas and Puran. Similarly, avla is native to India and has enormous diversity. They have potential of afforestation and carbon monetization, sequestration alongside the forest and fruit tree. Total area under fruit tree in India is around 27,476.1 million hectare and total production accounting around 334,602 metric tons according to NHP data 2021. Among these, the bale occupies an area of about 1.2 million hectare with the production of 82.26 metric tons, and avla occupies the area of about 100.48 million hectare and production of 1196.84 million tons. Bale, it is an underutilized crop. As you know, it is a highly nutritive and having medicinal property. Bale can thrive in a high altitude up to 1200 meters and can tolerate the temperature range of 4 degrees centigrade to 50 degrees centigrade without any significant growth retardation. During prolonged drought, fruiting may cease, but the plants can survive where the other plants cannot be survived. Suitable for alkaline, stony, and sodic soil, up to 29 ESP bale can survive, and with the pH range of 5 to 10, without application of chemical amendment, bale can be grown. Similarly, at CISH, we have collected lot of genetic diversity from UP, Bihar, MP, and Jharkhand, and Chhattisgarh. You see the range of variability available with the CISH. A total of 58 accessions which have been planted in the field gene bank were characterized, and the range. This is the phenology, morphological key characters of the different bale varieties. This is the fruit characters of different bale varieties. You see the fruit shape and size variation. And this is the range of we have evaluated the germplasm, and this is the range. You see the fruit weight vary from nearly 900 to 2.28 kgs. And in bale, mainly three things we are considering while we are characterizing the germplasm: then the fruit shape and size, and the weight of the fruit. Generally, we prefer from 1 kg to 1.5 kg of the fruit because bale is generally using as a for making sarbat, and bigger fruit size having their own problems. The yield is less, fruit drop is more, as well as the ripening of the bale or uniform ripening is another problem. So 1.5 to 1 to 1.5 kg fruits with thin cell, one nearly less than 2 mm thin cell we prefer. So these are the different these are the parameter and range of these after evaluation we have found these are the ranges. And one after thorough evaluation, one promising genotype, that is CISH Bale 3, were released at the institute level, and it is in the quadrated cell also. The chief character of this variety is dense canopy is nearly thornless and medium fruit size, and age with prolific period 56.27 kg at the age of 15 years. Fruit medium 1.15 to 1.5 kg and attractive golden yellow color. And moreover, it is tolerant to frost, relatively less food drop, and free from fruit cracking. These are the three attributes which differs from the other genotypes, because in Wales generally fruit cracking and fruit drop is a common problem. And we have compared with this variety with CISH B1, the fruit quality is comparable. Only thing is, you see, these are the characters. Otherwise, the B1 and B3 are almost similar in fruit characteristics. Only three is that it is tolerant to fruit cracking as well as less fruit drop and less frost injury. In Wales, generally during winter season, lot of injuries, leaf injury is there, leaf drops down and fruit also drops down. So this variety is little bit tolerant. So comparable with B1 as well as tolerant with these characters. So this variety was released at the institute level, and it is in the coordinated program also. You see this B1 and B3. The B1 having the cracking problem and B3 is free from cracking. Similarly, Amla also we have collected lot of genetic variability from the Madhya Pradesh, Gujarat, Rajasthan, Bihar region, and we have evaluated, and you see the variation in that variability, and these are the characters variability we have found, particularly the vitamin in Amla. Generally, we are searching the variety which is having high vitamin C as well as the phenol content, as well as the fruit weight. These are the three major parameters from which we are looking for, and these, on the basis of evaluation, we have found two selections. This is the fruit shape and size of the commercial variety. CISH Amla 1 we have identified and released at the institute level, and this variety is having the higher fruit size, fruit weight 40.65 grams, and higher vitamin C as compared to other variety. You see 500.9 mg per 100 gram of the pulp. Total phenol is also high and fruit is also high. So this variety we have released from the institute level. And second, we have compared with the Kanchan variety. This second is the Amla 2 variety. This is the Amla 2. This is the fruit of Amla 1, and this is the fruit of Amla 2. Amla 2 is the colored variety. Generally, colored Amla, we have found during the survey that if the fruit is the color, then the size is very small. But in this, around 20 to 25 gram fruits, we have fruit weight, and vitamin C content is also very high. You see 17.44, and total phenol is also high. So these are the two selections which we have released at the institute level, and these are in the ICRP program also. ना रीवा का नहीं, ये मध्य प्रदेश का ही है, पन्ना फॉरेस्ट एरिया से हमने कलेक्ट किया था मटेरियल, और ये दोनों मटेरियल जो हैं ये पन्ना सीधी शहडोल एक बड़ा सर्वे हम लोगों ने किया था, उससे जो मटेरियल निकाल के फिर ये जो दो आइडेंटिफाई किया है. तो in the nutshell we can conclude that these are the fruits and these are the variety which can be grown in where the climate change is taking place in those areas. Thanks.
Next speaker is Dr. R.K. Yadav. Dr. Yadav is from scientist at IIT. He was going to speak on the okra breeding in India, present status and future challenges. Dr. Yadav.
बना है पीपीटी आर के यादव ने ग्रेड 3 ये बाकी इसमें हुआ सब मेरा पॉइंटर कहाँ गया वर्किंग है अच्छा दोनों चीज़ आता ना तो good evening to everybody, respected chairman, co-chairman, reporters, distinguished scientists, all the participants, ladies and gentlemen, students. As we know that the okra is very important crop from India point of view because we are the largest producer of the okra in the world and our contribution is very high and in terms of production. Almost you can say more than two-thirds okra is produced in India only, and we are also exporter of okra. And the private sector in India now this they are playing very crucial role in popularizing okra because the okra has become a global crop for private sector from seed point of view, and nowadays it has surpassed the seed value of the other crops like tomato, brinjal, chili, etc. So now okra has become number one company. And some of the company, they are doing the business of more than 100 tons per year. Per year, they are selling of single variety. Some of the company, especially Advanta and this variety, this company. So okra has become a very important crop for India also. And we see that the okra, this is also called ladies finger because the shape is like that. And we are, you see that at present we are producing 63% of okra, and a lot of important uses are there. Out of that, very important use is like that. You can see that this is the only vegetables where we can find the iodine. So this is the source of iodine. Otherwise, you will get iodine from iodized salt only. So in the vegetables, the importance of this okra. Mucilage is also very important from industrial point of view. Lot of uses are there from this medicinal point of view, medicinal industry and chemical industry and food value also of this mucilage is very high and used as a additive material in many things. Economic value of okra is very high and it can be grown very important in different cropping system. You can see this in this figure that Asia we are having very only 25% area, 23% area under okra. But if you see the production, we are producing more than 65% of production. Our productivity is high. Means and Africa, very high area is there, but because of low productivity, and their production share is very low. If with the growing trend of this okra, this world level, we can see that more than 10 million, 10 million tons this okra is produced worldwide. Out of that, more than 6 million tons, exactly 6.3 million tons of share of the okra is from India only. So this much contribution is India, and this you can see this in the figure, and this also I have told, and this state-wise also this many states are growing okra in India, West Bengal, Gujarat, Odisha. These are the very lead player in okra, and many countries. If you see this availability of the okra in our country, that maximum time it is available from April to October, like summer only. But in some of the states like West Bengal, Odisha and Gujarat, it is produced almost round the year. When this okra we are getting this time, that is coming from Gujarat, especially the Byara region of the Gujarat, known for this one. That area is only in the country where the okra they are exporting to other area. From Nasik area also they are, I have seen that they are exporting from that area. And from Chhattisgarh also some area like Raipur nearby where temperature is mild in the winter season, that they are going and exporting. So this is the country-wide, different state-wide okra status. And okra export, I told that we are exporting also. We are number one exporter. And 28% export share goes to only to Singapore. Then other Arab countries we are exporting. And if you see the breeding methods, what we have used in the okra breeding in our country, that lot of conventional breeding method was used earlier. Some special techniques was mutation breeding, polyploidy breeding was used, and some varieties are developed. That is the beauty of the okra. In all sectors you will find the commercial variety. And biotechnological and genomic advances has not been taken so well placed, but now we are working on that one and using this molecular techniques also. to develop some of the information. And breeding objective, mostly the quality is a major one, and yield is another one. And another resistant in major criteria in okra, two diseases are very prominent. YBMD was earlier. Now ELCB is another disease which come, and that again that has become very serious disease. Many parts of the South India and North India and then sub pockets is coming. So this is also has become a major issue for okra improvement. And if you see this different variety are different by different institute, like in public sector, major institute like IRI, IHR, IIVR, this HISAR, HU HISAR, and this TAU, Ludhiana, they have been the major player of the okra breeding point of view, and they have developed a lot of breeding material also. If you see the hybrids also, and especially this IRI Pusa Makhali was developed before 60, and in 50s you can say, this variety was developed, and Pusa Sauni was developed. This was a revolution in the country when Pusa Sauni was developed, this first variety, and a lot of hybrids are also we have developed from IRI and other places. And if you see the strength, weaknesses, etc. in this crop, the strength is that we can grow round the year, improve variety and hybrids are available with us. That is our main strength. But certain threats are also that they go virus or abiotic stresses. They will be the future threat for okra improvement. So if you see this okra improvement, this Dr. Harbhajan Singh was the pioneer breeder in the okra, and he has started the breeding of okra in our country, and he has developed this Pusa Makri, this first variety. Then later on he crossed some genotypes, and they have developed first YBMB resistant variety, that's Pusa Sauni. So that Pusa Sauni development has started development of many seed companies in our country, especially the micro seed company, which they started their business from this Pusa Sauni only. They have started their business with 2 kg seeds, and now you see the status of the company. So this was the contribution of this variety, and Dr. Harbhajan Singh was he knows the Indian background because he has not introduced only this variety. Lot of exotic material that time, around 50s, 60s, he started. And then some of the wild species, especially the Pusa Hebin mascot species, this many hearts, species many hearts, he has introduced from Ghana. That was my variety later on used by other breeders, other institutions in development of this YBMB resistant variety. So that's why, and because of he was the head of this plant development scheme at IRI. Later on, that scheme led to the foundation for the NBPGR that became on that is the same institute. So in the pure line, I will not go so much that a lot of material has been developed, and this hybridization has been done in this one. And I told that different institutes like Barsa Upahar and the Hisar Unnat, very 2 varieties and developed by Dr. B.S. Dhankar. He has also a pioneer breeder in the okra, apart from the Dr. Harbhajan Singh. Then we have developed one variety, the Pusa Bhindi 5, that is resistant to YBMB, and we have commercialized. Because you see in the beginning, this Pusa Sauni was commercialized. Similarly, we have started the procedure to commercialize this variety because in public sector, we don't have much institution like only NSC there, and we have given to private sector also, and some model we have developed so that variety should go to the private sector also and go in the farmer's field. So because that why we have developed one-time licensing, not a royalty every time. So one-time licensing was better. We found that rather than going for the royalty on the sale, one-time licensing and one-time license fee we are taking, and then we are supplying the material to anybody. And now we have developed Pusa Lal Bhindi 1 because the last 2 years the red okra has become very popular nowadays. So in keeping that was because this is a rich source of anthocyanin. So this variety earlier also Pusa Lalima was developed from IIVR. So but this variety has 2 times IO. They are containing the anthocyanin 165 milli, this microgram per gram or 65 ppm. We are this material that is 130 ppm, but almost double this one. And Pusa okra hybrid 1 that was also notified, and this was having dual resistance. YB this Pusa Sauni was this Pusa Bhindi 5 having YBMB resistant, but this hybrid having dual resistance. We have transfer in both the parent, and we have one YBMB resistant and another is the ELCB resistant. So dual resistance in this variety. Then interspecific hybridization we can see that has been used very widely in the okra, and lot of variety has been developed to interspecifically. So majority of in many crops it has not been used, but in okra this has been used. So lot of wild species are there; they have been used. and this from Parbhani, this Parbhani Kranti was developed, and many varieties from IHR, Arkan Anika was one variety. It was used for interspecific hybridization, so that was also leading variety. And if you see this genetic male study system to economize the seed production in okra, the genetic male GMS system has been used, but because of the certain problem in GMS, 50% male and female male style and fertile plants are there in. So that's why this system is not so much used. The private sector, so many varieties has been developed in private sector also. Mutation breeding, I told that in the beginning that this has been with many Punjab 8 and Parbhani Tillu MD has been used. This polyploidy is used, and trait specific breeding now we are doing for different traits. And one trait that is a very close internodal plant idiotype that is very becoming popular nowadays. So because of that, one variety has come. Because of that, all the okra breeding system has changed. To go for tall varieties, they have gone to short variety with closer fruitings. And this is the quality and processing test. We will not go much. In this biotechnological aspect, we can see that a lot of molecular work has been done in okra, but still we have many, not as because the genome size is very large and chromosome number is very large in okra, because mucilage is another this problem for extraction of DNA. So that's why many people have not come, but we are starting, and very international level also some at ABRDC people are doing this one. So these are the major challenges breeding for weather resistant in okra, because this is the most of the varieties they were monogenic resistant. So breakdown of the resistant was there. So this we will try to for longer duration resistant. Then pre and zygotic barriers are there in okra. We will go here and we will go that breeding of durable resistance. We have to go for novel source of different sources like tetraphilus has been used, many heart has been used. But one is source of this evil mascus, mostatus, we are using that one. So that will be helpful in the development of this another type of resistance, long duration resistance. And you can see the pre and post zygotic barriers. We are trying to do polyploidy breeding and utilization of some other, we are trying resumption protocols. And here we have started utilizing resumption protocol. And limited use of biological that I approve, that genome size and etc. because of there. And we have developed, because in the transport data is not available in okra, markers are not available in okra, very few markers are there. So we have tried to develop through transcriptome, this YBMD link marker. So we are trying in that process, and we have produced. So we will not go. Thank you very much.
Thank you. Thank you very much. Thank you. Now, this one we have completed the innovative lecture. Now, we are going to start this oral presentation. In oral presentation, there is provision for the award. Therefore, I request the presenter to be strict with your time. Time allotted for each speaker is only 5 minutes. And 1 minute for the discussion if required. No introduction, nothing is required. State away what you have done, what you want to communicate. So very straightforward. The time is 5 minutes. Keeping that time limit, I would like to call on the first speaker for the oral presentation, Dr. Anju Paul. Dr. Anju Paul is going to speak on performance of Tahiyya cultivar under the right region of Uttarakhand. Others will also be ready. Next speaker is Archana Mahida. Archana is here. Dr. Ashish Yadav, Dr. Siva Vedwal, Sunil Kumar, Dr. Tibyani, Dr. M. Patel, and Dr. Swapnil Pandey.
Very good evening, dignitaries and participants. Myself, I am presenting the paper performance of the cultivar in the Sarai region of Uttarakhand. I would like to introduce the crop, as this is the first work which I had done in this university, Introduction to Dahelia. Dahelia is a crop. It is a scientific name is Dahelia variabilis, and belonging to family Asteraceae, as we all floriculturists know about that. Then chromosome number is 2 and this, and Dahelia is generally a crop which is growing, and nowadays it is highly popularized as a decorative plant and pot culture, because we are having the limited space and we are growing these generally flower crops in pots. So this is the best crop for pot culture. Then antibiotics, anti-mycotic properties are also there. It is used for wound healing also from ancient time. And tubers serve as a source of fructose and starch, mainly used for earlier ancient time. It is for the animal feed, used as animal feed. And orange dye is also obtained from the bloom, and which is very much used for the developing the dye. Then the materials, we are having 11 genotypes which we are pursuing here with the 3 replications. This is the, and spacing was considered 60 and 40, and it is planted in December 20th. And this is the pictorial view of the experiment. This is the layout and the genotype under the study. These are Anadinath, Gyanijal Singh, Kenya Orange, Hiranmoy, Jishu, Mutangani. These are the staking which the work I have done. Then the experimental result. The plant is spread at different growth stages and different Dahelia genotypes. Generally, as the picture shows, the maximum plant spread was observed in tangent varieties. It is 19.08 at 30 days after transplanting. Then after 60 days, and maximum was considered in the tangent type of variety, then followed by the Kenya orange. Is then least was found in the Sachin plant spread. एक मिनट आना, बार में कर रही हूँ। अच्छा, इससे
कोई बात नहीं, आप बताइए पहले।
छोटा number of branches at different growth stages in different Dahelia genotypes. It is observed that maximum was found in tangent 2.85 and least was in Sachin varieties, but the Jishu and Kenya orange was little bit at par. Then stem girth, it was found maximum in case of Jishu at all the 30, 60 and 90 days after transplanting. Then Sachin was observed the least one. Then number of leaves at the growth stage of Dahelia, the maximum was found in Kenya orange in all these days after transplanting stages. Then Sachin was the least number of leaves having recorded. Then internodal length at different growth stages and the leaf area in different Dahelias. The internodal length was maximum in case of tangent and least was observed in the Hiranmoy. In the same case, the leaf area was observed maximum in case of Mutangani and least one with the Sachin. Then the flowering parameter, as it is a decorative flower which we can grow in the bed area and pot area. So the flower characters are also very important for this crop. The maximum days to bud appearance was observed in case of Sachin, means it is delay type of varieties, and days to flowering taken in the Sachin 119, and maximum duration of flowering was observed. Okay. These are the estimated variance and genetic variabilities with which we can correlate. The maximum genetic variability was found in the flower weight, and GCV genetic variability also in the flower weight category. In this, I would like to conclude this that the Mutangani and the Hiranmoe variety was found the best one for the pot culture, and Sachin was considered as a delayed crop or least performing in this climate. Thank you.
Okay. Next speaker, Ranu is going to speak on application of PHC marker gene to the genes for late blight and potato cyst nematode stand in indigenous potato genotype.
Folder. Yes. Yeah. Open it. In this, yes, please. Yes. Respected dignitaries and fellow participants, a very good evening. My topic for today's presentation is application of DNA markers linked to the genes for late blight and potato cyst nematode in indigenous potato genotypes. I wrote the wrong technical section due to some errors. Okay. Potato, a versatile humble crop, belong to family Solanaceae, and the polity level is from truly diploid to the hexaploid one. The origin of this crop is not in India, in the lake Titicaca in South America, and it was introduced just only 300 or 400 back to our country. And if we say potato is not the origin of our country, what is the pre-bred CPRA varieties? These are actually the initial potatoes that were brought in our country, and they are the chance seedling survivors of this. And they are having very specific, peculiar good qualities like dormancy. They are having the high temperature tolerance and very good culinary qualities, and still they are popular in very remote pockets of our country. So coming to the biotic stresses, which is the topic of my talk, potato suffers from a variety of biotic and abiotic stresses. Among that, the late blight is very peculiar, and still it is popular, known as the Irish famine, and still it is prevalent both in the temperate as well as the tropical part, subtropical part of our country where the potato is grown. So there is a need of at least the highly resistant to resistant varieties for the hill breeding, while the moderately resistant varieties for the subtropical where the 90% of potato is cultivated. Second is the potato cyst nematode. Two species very important, Globodera rostrokinesis, and another is the Globodera pellata. An important quarantine pest of potato world over and across the world as well as in our country. And recently, this pest has been intercepted in the northwestern hills, including the Himachal Pradesh, Jammu Kashmir, and Uttarakhand. As a result of this, the whole seed industry of this area has been devastated. We are not able to produce a single seed of potato, and we can see the cyst of this potato cyst nematode, which is actually a female body, which constitutes nearly 300 to 400 eggs. And this particular cyst, it can survive in the soil in the absence of potato crop up to 20 to 30 years. And we can see the Gazette of Government of India in 2018, of which we are not able to send even a single seed of potato from our hill location to different parts of the hilly locations of our country. So that is the gravity of these two pests. Now what we are doing from CPRA past so many years, we definitely were having this in our mind that these are the devastating pests. We came with a variety Kufri Karan in 2020. That is the first marker-assisted breeding variety of potato in our country, which is highly resistant to late blight as well as moderately resistant to potato cyst nematode. But moderate resistance is not the answer. We need a variety which is highly resistant or immune. So we came with one more advanced clone of potato, that is SM11120, that we have registered in NVPGR with the IC number, but still this advanced clone is having certain drawbacks, and we are trying to mitigate it with the agronomy. So this is just a glimpse of how we are managing this potato cyst nematode and late blight in case of potato. Now coming to the material methods of my study, I took 72 accessions of native this potato accessions that were cultivated in the Kufri region, as well as they are conserved in vitro in the tissue culture. For the late blight evaluation, we did for the 2 years in the Kufri hills, which is the hotspot for potato late blight. And this is done according to the AUDPC value. And in table, we can see how the genotypes, they are categorized according to different AUDPC value. That is the area under disease progress curve. While for PCN evaluation, we followed the root ball technique. It's a very popular technique by the Krishna Prasad et al. And similarly, the genotypes, they were categorized according to the highly resistant to highly susceptible, according to a table we can see in this slide. While molecular analysis, the standard protocols, they were followed, the published markers, they were used for the analysis. These are the details of markers for late blight. Different RGs, they are controlling the resistance for late blight. It is a quantitative resistance. We have used 3 RGs, that is the R1, R2, R3 for late blight evaluation, while for PCN, for the rostokinensis species, we use the marker TG689 and 57R that controls the H1 gene, while GRO141 that controls the GRO14 gene. While for Globodera pallida, it is the SCN spud marker we used. If we see the phenotypic results, majority of the genotypes, they were fall in the susceptible or highly susceptible. Only 2 were resistant, while 2 were highly resistant. While for PCNs, the highly resistant accessions were 3 each for Globodera, rostokinensis, and pallida, while only single accession was resistant for Globodera pallida. We can see this in this. Out of the different markers for late blight for R1, there were only 11 accessions out of 72 that were having the marker R1 that is controlled by the COS8 gene. While for Globodera rostokinensis, 3 accessions were having the TG619, 5 were having the 57R, and only a single accession is having the GRO14 gene at 602 base pair. While for Globodera pallida, that is controlled by the SC276 marker, 64 accessions, they were showing the positive results. Now coming to the conclusion of my study, different accessions, native accessions, they were studied in this analysis. We found one accession, JG1, a peculiar accession from the East Khasi region of our country that is highly resistant to late blight phenotypically, and it is also highly resistant to the Globodera rostokinensis and Globodera pallida, and it is also confirmed by the markers. There is another indigenous accession, garlantic. It is a collection from the Kanpur, Uttar Pradesh. It is highly resistant to only the PCN, both Globodera rostokinensis and Globodera pallida, and it is confirmed through markers also. Jivan Jyoti, a good variety from Bardhaman, West Bengal, a hub for the potato cultivation. It is highly resistant to both the species of potato cyst nematode, and this was confirmed by the markers also. Kanpuria Safed, as the name depicts, it's a indigenous collection from the Kanpur, Uttar Pradesh, and it is highly resistant to only late blight and confirmed also to the markers. While Rangpuria, another collection from the Kuch Bihar region of West Bengal, it is highly resistant to late blight, and it was confirmed to the markers also. Now coming to the conclusion of my study, like we have studied the different indigenous accessions, which are the base for the future resistant breeding for a potato, particularly for the northwestern hills. And also, like these varieties, they are very popular in remote pockets, even though, so that we can supply the good quality seed material that are disease-free for these locations, so that farmer can be benefited. Also, different molecular studies can be done, especially for the GRO14 gene, which was actually absent till now, whatever the accession we have studied for in potato for potato system. So this is the aerial view of the location. Thank you. Thank you very much.
Thank you. Very nice to listen. Now next speaker is Archana Mahida. She is going to speak on effect of fortification of iron and zinc fertilization on quality parameters of mango cultivation.
Thank you. Thank you. Forward and this is going forward.
Good evening to all. Myself, Archana Mahida. My oral presentation title is Effect of Fortification of Iron and Zinc Fertilization on Quality Parameters of Mango Cultivar Kesar. Micronutrients play major role in crop production and disease resistance. And so I decided to take this topic that zinc and iron occupy the important place due to Zn is its availability to positive influence on the plant growth and development, and iron plays an important role to promote starch formation and cell enlargement. And there are the 3 different methods for fortification. First one is fortification, the second one is convention breeding, and third one is spatial method. And among them, I selected the first one, that is fortification, that is agronomical practices. And I choose my Kesar variety because the Kesar has found good yield potential almost a regular beer mid-season variety and having a good consumer acceptance and attractive shape and size. And I selected foliar spray of micronutrients, that is iron and zinc. And this experiment was conducted at NAU Navsari on 20 years old mango orchard by the spacing of 10 by 5 meter distance at SP College of Horticulture and Forestry, NAU Navsari, and conducted in CRD design with 9 treatment and 3 replications. And there is these are the treatments. The foliar spray of alpine and Zarden at the time of application is flowering, P and X stage of mango. And these are the quality parameters I have.
Archana, come on, major findings and come to the not required the detail of the material.
And sir, from this research, I found that T9 treatment, that is 0.50% of FeSO4 and 0.50% of ZnSO4, found superior among all these treatments. It found that 90.35 bricks of TSS, 36.70 mg per 100 gram ascorbic acid, and there is no significant result found in organoleptic score and shelf life of mango. And this might be happen due to hydrolysis of complex polysaccharide into simple sugars, synthesis of metabolites, and rapid translocation of photosynthesis products and millets from other part of plant or to developing fruits. And ascorbic acid also recorded significantly higher in T9 treatment, that is 0.50% of FeSO4 and 0.50% of ZnSO4, because it might be happen due to ascorbic due to catalytic activity of enzyme and coenzyme which is involved in ascorbic acid synthesis.
So, next speaker is Dr. Asif Yadav. Dr. Yadav is from Central Institute for Horticulture Control, and he is going to speak on utilization of exotic varieties in mango.
Good evening, sir. Namaskar सभी लोगों को. We are working on mango since 1975. I am continuing the work done by all the legendary personalities, especially Dr. Rajan sir. Mango is the most important crop of the India, and we are exporting mango to more than 40 countries in the world. And especially we are using these exotic varieties for their various traits in the development of new varieties. And we have 18 exotic germplasm in our collection. This Tommy Atkins, Sensation, Edward, Eldon, Keat, and many others. Some of the important characteristics these varieties they have good fungus resistance, very long shelf life, and good handling characteristics. That's why Tommy Atkins is the most important variety of export potential in US. And it is there are many other varieties like Sensation. We are using this as a donor parent because of high yielding and color combinations. Other varieties like Edward, they are not very good for us. Some of the varieties like Austin, this is also very good variety, and it is very good performing in our subtropical region for very high yield and very large food and very high pulp ratio. I missed Kensington. Kensington is also very important variety for Australia. We are also using it as a male parent, as a donor parent. Maya is one of the very good variety of Israel. It is like apple in shape and very sweet, very sweet. These are the photographs: Tommy Atkins, Keat, Maya, Kent, Sensation, Kensington Pride, Austin, Kitchener, Pradium Racia. We are also observing the performance of these exotic varieties in our conditions.
especially Tommy Atkins, Kensington Pride, Austin. They are very good and they are very much better in our conditions. And fruit size is also more than 500 grams. Pulp percent is almost 70%. And TSS is more than 20 degree bricks in many of the varieties. And our improvement program started in 1975. And we mainly focused on red field varieties with lesser sweetness right now. Good sugar acid blend. These are our the focused area. Very long shelf life we are looking for. That is good for export purpose. At present, we have lots of progenies in our pipeline. Of progeny 1739 is under the ICRP trial, but is very late maturing and very good shelf life. So this is a very good, and it is performing very well in many of the centers. Other varieties, other hybrids, they are in the pipeline. This is 4065. It is very good, very attractive color, and TSS is also very good. We have also worked out the nutraceutical parameters. Many of them, they are performing. Besides regular bearing and high yield characteristics, we are also focusing on the nutraceutical properties. Many of them, they are having very good potential. We have also worked on the manufacturing in luteol and carotenoid content, and most of them, like hybrid 1042 and 3669, they are very good in nutraceutical potential. So we are also working on the biofortified varieties. And my conclusion is that why we are using the exotic varieties, because they have very good adaptation worldwide, and they have originated in India and they are performing very well in our place. So we are transferring the adaptability characteristic here, and regular bearing and red fleshed. Thank you so much.
Thank you, Ravi. So now we see Siva Bilwa is here. So Siva is going to speak on important morphological markers obtained in Gladiolus variety since the past.
Good evening, everyone, all the respected dignitaries and my dear friends and juniors. I'm here to speak about my topic, important morphological markers that I've obtained for characterization of mutants in Gladiolus variety Prince of Orange. Out of the 3 varieties that I've chosen to study for my mutation trial, this is one of the variety in which I found significant changes. And the objective of my study was to first induce the mutations in my variety, work out the effective dose, so that the dose that is bringing out the most changes so that I can work out the effective changes in order to bring out the most effective dose in my experiment and to screen out the useful variants that I have obtained. For the material method, I have chosen 2 mutagens, that is gamma irradiation and ethyl methane sulfonate, one being a physical mutagen and another being a chemical mutagen. And I had chosen total 360 comps for my study. The gamma irradiation doses varied from 4 kilorad to 5.5 kilorad, beyond which the lethal dose was obtained, so we did not take it further. And the EMS was chosen from 0.2% to 0.6%. And some of the variations that I found in gamma irradiation was that you can see that the control, like if we compare the control, we can see that in 4 kilorad radiation, a lesser number of florets was found, and smaller plant height was obtained with shorter bloom duration. So we can say that we can use this kind of variation as a pot purposes. And for 4.4 kilorad radiation, I obtained that color change from orange to maroon color, and it has been stable for 2 consecutive generations. So we can say that it is a promising mutant of this particular control. And similarly, I found smaller plant height and wedges height and shorter bloom duration. In 5 kilorad dosage, I found white patches appear on the petals. Again, this was a stable mutant. And in 5.5 kilorad, I found purple blotches with white spots on the petals, which is again stable. Then in EMS, I found really good quality parameters in my crop. That is, the number of florets increased. That you can see in control, the number of florets are around 10 to 12, which increased from 18 to 21. And this had more waste life as compared to the control. The control was only for 11 days. While this, the mutants, the waste life increased to approximately 22 days, which is a good study for the post-harvest parameters, because in floriculture, for commercial floriculture, waste life is a very important parameter. Then we can see the length differences. The first plant being the control, then 4.5, 5 kilo red, 5.5, and so on. The EMS doses, we can see the variations in length obtained, and these are the various parameters. And from these parameters, I would like to conclude that from the physical and chemical mutagen that I undertook for my study, chemical mutagen proved to be very good as far as quality parameters was considered, but physical mutagen showed more changes in terms of length, floral qualities like length, colors, etc. Thank you.
Now, next speaker. Sunil Kumar. Sunil Kumar, he will speak on gamma radiation induced mutagenesis in peach.
Honorable Chairman, sir, respected members of the judging committee, scientists, and my dear friends. I am presenting my small piece of work, that is gamma radiation induced mutagenesis in lychee. We know that lychee is very important fruit crop that is popularly known as the queen of fruits, and it is the demand of lychee is increasing day by day in national and international market. But the yield of lychee is very low as compared to its potential yield. So one of the probable reasons for low yield is the lack of the superior variety, because lychee in India is mostly dominated by the Sai and China variety, and they are suffering from one or more defects. Okay, so varietal improvement program in perennial fruit crops like lychee is very difficult through the conventional breeding. So mutagenesis to induce genetic variability in lychee was started last year. So the methodology was used. Lychee cultivar Sai was selected for, and the fresh seeds were exported with different doses of gamma irradiation, that is 5 to 50 gray of the gamma irradiation at IRA New Delhi. A total number of 1,000 seeds were treated, 100 seeds in each treatment. And the treated seeds along with the wild type were then sown in the root thinner, and after treating, seedlings were transplanted in polybags, and they were observed for their survival percentage. So this is the slide shows the glimpses of the mutation work in lychee. So results that as lower dosage, the results were inconsistent, but if you see the dosage at 45 to 50 gray, there was a reduction in the survival percentage. So it initially it highlights that the dosage 45 to 50 gray are the have the potential to generate the much needed variation with lychee. Thank you.
Very good. This kind of speaker we want. Next speaker is Dr. Sidyani from IRI. He is going to speak on the mediated immunogenesis theory of outer integuments. It's mediated, synchronized, indirect somatic immunogenesis in chinomembrane.
So, a warm welcome to all the scientific community present here. Myself, Devanai. I have worked on in vitro mutagenesis under the guidance of Professor Om Prakash Avasti. A part of my research work, that is standardization of protocol for regeneration, resulted in indirect somatic embryogenesis. It is of uniqueness, some kind of uniqueness was present in this particular protocol. So I have thought to present as an oral presentation. And Kinno, it is an introduced interspecific hybrid, and it has revolutionized the citrus industry of northwestern India, and because of its production and quality. But the commercial utility was hindered presence of high number of seeds. Hence, the conventional mutation breeding, that is budded irradiation, was initiated at IRI by my chairman during 2011, but the improvement was delayed because of occurrence of chimera. Hence, unicellular genetic modifications using tissue culture have a potential in this regard. So we have initiated in vitro mutagenesis. We wanted to initiate it in vitro mutagenesis, hence the particular work was proposed. Our major objective was production of synchronized, low-cost, indirect somatic embryogenesis system with year-round experimentation. It should be helpful for year-round experimentation, and also it should result in single-cell regeneration. And the materials and methods were, the plant material which is chosen was in the outer integument derived embryogenic calli, and the methods followed were, the callus will be subjected to habituation, then suspension culture initiation, followed by somatic embryo initiation, and then it will be subjected to maturation, germination and conversion, followed by establishment and hardening, then molecular and microscopic validation. And based on our preliminary work, we have chosen 15 treatments, and the results are, since species-specific response was there in citrus, we have undergone systematic investigation with different explants, starting from, it is by representation of juvenile, reproductive and vegetative stages. Among that, the fruits which is undergoing, the fruits of color break stage, where the seeds will be of having fully matured, but it will not be having it will not be desiccated. At that stage, when we are giving incision at the microcallosal end, it is having ability to produce embryogenic callus. This callus was subjected for habituation, that is nothing but growth regulator free medium. We have to maintain this callus for 6 months. Then it was, when we are subjecting this callus after 6 months by frequent subculturing 2 times, then it will be ready for suspension by having active callus growth. And this is the different treatments which is showing the suspension mediated multiplication of callus. And among the different treatments, SSM1, which is which had a highest somatic embryogenesis ability within short duration. And this is the photographic showing SSM1 with a somatic embryogenesis and different stages of somatic embryogenesis from globular to hard stage. And similarly, when we are subjected to maturation and germination, where maturation and days to maturation was early at SSM15 and germination was early at SSM12. And these are the different stages showing maturation and germination. And after germination, bipolar conversion was initiated. Then they were subjected to hormone-free media for establishment. Then hardening was achieved on cocopeat, vermiculite, and perlite combination of 2:1:1. And then we have followed. We have subjected these planting materials what we have obtained for genetic fidelity testing, where we have chosen CRD design. We have chosen 6 randomly selected plant material along 5 randomly selected planting material along with mother plant, and 8 different ISSR markers were analyzed. Among the 2 markers have able to identify some kind of variation. And single cell regeneration, assessment of single cell regeneration through microscopic studies, where after suspension culture of 14 days, they were having smaller clumps of 3 to 4 cell on single cell also. And after 20th day, they are having globular to arch phase structure. And the summary is, embryogenic calli can be initiated from outer integument at the transition from the seeds, when it is having transition from fully maturation to desiccation on MS medium, which is supplemented with activated charcoal 200 milligrams. And this calli, when it was subjected to habituation, it is having stability. And when it is having stability, it can be maintained for longer duration. And also, since it is maintained on the hormone-free media, it is having very less variation than other reports. And also, when it is subjected to suspension culture on hormone-free media, it is having 92% of somatic embryogenesis within 21 days, and same has had a maturity on empty medium supplemented with coconut butter 10% and NAA 0.5 milligram, and it had a higher germination on DKW medium with carnitine 5 milligram and malt extract 1000 milligram. And the establishment was again subjected to growth regulator free medium on 87%, and the final recovery was around 70%, which is higher than other reports. And ISR markers had distinguished the plants for identifying the variability. And from this, I can conclude that this is an alternative protocol for single cell regeneration, and it can be efficiently utilized for in vitro mutagenesis. And this is the entire protocol. And the uniqueness of this study is it is not reported, means outer integument derived embryogenic callus is not reported seriously. And since habituation has been done, this can be utilized for longer duration. Has a genetic material, we can store it, and this can help us for year-round experimentation. So, and since growth regulator free medium and only 20-day suspension has reduced the cost of production, and it is very simplified, and a high plant rate recovery will be there, and it is efficient alternative. Thank you.
Thank you. Next speaker is Dr. Ajay Patel. Dr. Patel is going to speak on the evaluation of local personnel of southern Gujarat region.
Good afternoon, one and all. Myself, Mukesh Patel, working as an assistant professor in Asli College of Horticulture. My topic is collection and evaluation of local germplasm species around South Gujarat region. The turf grass, generally, we know that generally it is used as a sports, also as a aesthetic purpose, particularly. And in most part of the India, particularly the selection one, that is Cynodon dactylon, is widely used as a aesthetic turf as well as spot such turf. So our present topic was selected to find out the local germplasms that are comparable with this selection one. So we have collected 9 different germplasms around the our South Gujarat, and we have compared it with the selection one, that is Bermuda selection one, and the design was randomized block design with 3 replication and 2x2 gross plot size and net plot size of 1.5 by 1.5. This is the general layout of the our experiment. These are the different turf. Actually, we have collected 9 different turfs, and among them 2 were not found. They are very poor in performance. So we have removed with the suggestion of statistician. So the G5 turf. This is the selection one turf, which is widely used in sports as well as utility purpose. Among them, our result shows that the G5 turf shows the that is selection one shows the highest leaf bed good texture. That is compared, while the G9, that is showed the leaf blade texture, that is 3. In case of establishment rate, actually, when it is the counted the number of days within that, it covers the 90% of the area, and we found that particularly the lowest day for 90% coverage was taken by G4 turf, which is at par with G9, that is 31 days. In case of aesthetic appearance, on the rating of 1 to 5, around their uniform spread, we found that G5, that is the hybrid selection one, recorded the best performance, which was at par with G9, that shows in full analysis, that shows the 4.38 rating. In case of turf quality, again G5, that is the hybrid selection one, recorded the highest rating, which was also at par with G9 turf, that is the local turf. In case of turf intensity, again G5 recorded the highest turf intensity rate, which was followed by G9. In case of turfing ability, again this G5 turf recorded the highest toughing ability, but which it was at par with our local tough grass species, that is G9. And in case of root depth, the highest root depth was recorded by G7 and highest recorded by G2, followed by which was at par with G7. In case of shoot root ratio, the lowest shoot root ratio was recorded by G10 tough and which was at par with G9. This is the picture shows the root depth of different tough grasses, that is G5 having that you can compare that the selection one and the root depth of our local tough grasses, and among them G9 recorded the very good root depth. So from this conclusion regarding its aesthetic appearance and all other characters, G9 tough can be used as a local tough, and it is having a good aesthetic appearance and also having a good wear and tear capability compared to G5 tough, that is selection one. So it can be used as a it can be used and further explored for different uses. Thank you.
Thank you. Another presentation is again from Gujarat. Dr. D.S. Mishra, he is principal scientist working at CIE Vijaipur. He is going to speak on phenotypic diversity of red plus guava with special reference to Western India.
Okay, thank you, sir. Respected chair, open group. Okay. Respected chair, Dr. P.L. Saroj sir, co-chair, Dr. Hazarika sahab, reporter, Dr. Verma, and distinguished scientists sitting in the hall. I am going to present phenotypic diversity of diversity for food quality traits and bioactive compounds in red fresh guava. So this guava need no introduction. It's a hardy species and had various uses. These are the various products can be made from the red fresh guava. It is good for healthy heart, anti-diabetic, lowers risk of cancer, vitamin C rich fruit, that is why it enhances immunity. And there are so many things are written in introduction, and my real research details can be downloaded from this. These are the list of guava genotypes used in this study, and these are the descriptive statistics for tree growth, fruit yield, and seed traits in guava genotypes. There was wide variability for these traits, and this HSD24 was having high tree volume, and this HSD15 was having more number of fruit per tree, and yield was also high. But this was also good. This was also having less 100 seed bed and seed hardness. Next, there was also wider variability for food chemical properties in the guava genotypes were studied, and amongst genotypes, the highest between TSS and titabilistic ratio was found in case of HSG15. It was also having some higher amount of vitamin C, along with this H1, which was having higher amount of acid and along with vitamin C, and it was having also having higher amount of non-reducing sugar. And this H4/3 was having more pectin content. Then we also studied quantified the amount of minerals and bioactive compounds present in the flesh of foods. This H1 was having more amount of potassium, potassium along with beta carotene, and it was also having more amount of total antioxidant activity. Principal component was also studied to delineate how the different components which were studied, how they are affecting the genetic variability. So principal component one was having strong association with traits accounting for antioxidant value of guava food. For example, beta carotene, total phenol, and total antioxidant activity. Similarly, PC2 was found associated with yield component, and PC3 was found. PC4 was found to be related with total soluble solids in red plus guava along with sugars. Then PSM correlation studies are also done to between major trades. Fruit per tree was having positive correlation with yield per tree. Fruit length and width was having positive correlation with fruit weight. 100 seed weight was showed positive correlation with seed hardness. Likewise, there was positive correlation with total phenol with total antioxidant activity. Flagnot also showed positive correlation with total antioxidant activity. Calcium had positive correlation with magnesium, while potassium had negative correlation with calcium. These network graphs modeling showing the same interactions amongst major trades. Clustering was also done. There were 3 clusters to form. H1 and H2 was most distinct because they were classed with their office strings of I and SG15, and group with SG15, 16, and 21, and others were in third group. In conclusion, we can say present study is that the red plus guava genotypes are remarkably rich in nutritional and antioxidant components. These genotypes we not only characterize but also use in different breeding programs for getting some good results. And these are the pics of some of the study genotypes, and these are some of the pics which we used in breeding program, and these are the results in terms of fruit shape, pulp color, and quality. So we did not only characterize, we also used in breeding program. That was the objective, to use them in breeding program to produce nutri-rich varieties, which is the need of the hour. Thank you very much.
Thank you. So the last presentation of this study, Dr. Sukhil Pandey. Sukhil Pandey is going to speak on phenotypic and biochemical evaluation of apple cultivars in Kandi region of Punjab.
So good evening, one and all present here. Today I'm here to present a piece of work which has been conducted at our regional research station of Punjab Agriculture University, Ludhiana, on phenotypic and biochemical evaluation of apple cultivars in the Kandi region of Punjab. So Kandi region means it is the region which is along the lower Shivalik hills of the Punjab, and this region has a problem of soil erosion, low water availability, and poor quality of soil. So as we all know that apples, these are a rich source of various phytochemicals that include flavonoids and other phenolic compounds, and these are responsible for boosting our immunity. And presently there are more than 7,500 of cultivars which are widely grown in each and every part of the country. And most of the variety of apples, when we think of apple, we think of a red apple. And these varieties, these are confined to the areas which are having 500 to 1000 of chilling hours, means low temperature below 7 degrees Celsius. So there are very few varieties of apples that are being confined to the conditions where the weather condition is mild, that is chill hours below 500 hours. So some of the chilling varieties, low chill varieties are Anna, Tropical Beauty, Early Fuji, Dorset Golden, etc. And the presence of these low chill variety widens the scope of apple cultivars in the areas having mild climate, basically the subtropical regions of our country. So the experiment, it has been conducted at the fruit research farm of RRS Ballowal Sonkdi. It is near SBS Nagar. And the statistical design was a randomized block design. And in the experiment, we have taken 31 apple cultivars, and these cultivars, they are grouped as low chill, mid chill, and high chill varieties. And among these, only 2 cultivars, Anna and Golden Dorset, have came into flowering and fruiting. So I'm going to present the physicochemical properties of these two varieties only. So it's a 2-year data, and upon the physical analysis, we have observed that the fruit weight, fruit size, and fruit yield, it has been observed more, maximum in Anna as compared to Golden Dorset, and these are significantly different from each other. And as you can see, this is the phenotypic difference among both the varieties. The Anna, it has some red marks, while the Golden Dorset, it is slightly yellowish green in color. Upon the physicochemical analysis, the firmness was more in Anna. The TSS acid-free and TSS acid ratio was also more in this Anna, but these were non-significantly different from each other. So I conclude the experiment that among the low chill cultivars which have been grown at our station, Anna has been found superior in respect to all the physicochemical characteristics, and also the PAU has recommended these two varieties at a small scale plantation, as in just like your kitchen garden, you can plant these two cultivars. Thank you.
Thank you. So this way we have completed the session, crop improvement and in and I promised earlier that there is any question. There is question in 1 minute. Please ask. Any question, any speaker, important question, not another statement, not important, not any question. Yes. But Anna planting is a very no, sir, they are so it is already we are there. So just few words, just few words I would like to communicate. That this in this session, there were 10 listed speakers as a invited speaker, and also after 9 turned up. And in case of oral speakers, there were 23 listed representatives, but only 10 had turned up. Overall presentation was very nice. Particularly, I would like to appreciate the young scientists who have presented briefly in very exciting 5-minute time. Generally, it is very difficult to express your views in 5-minute time because mentally you have prepared for more time. But anyway, all speakers here thank. As far as invited speakers are concerned, really it was very informative, very interesting, very good information was there. But I personally apologize that the time allotted was very less. So in future we will plan that the number of speakers we will reduce, but the time it will be very increased. The first speaker, Dr. Sandhya Gupta, he had spoken very well about the conservation strategy and given a very vivid account of how the germplasm can be maintained both at the field game bank and at the crop production. He has also expressed the need of germplasm. The second speaker was Dr. P.K. Dubey, who was from CPVFRA, and he has expressed the need of the protection of the plant variety. Many acts and provisions he has explained here, and something really interesting. And if anybody is interested, certainly you can ask to them if some queries there. Dr. R.M. Sharma, who has presented about the Indian scenario of the walnut. Walnut is a really important crop, export-oriented crop. And he has talked about many aspects of the walnut cultivation. Interesting concern was that our contribution in the global scenario of the walnut is just 10%. It really needs a matter of concern and is required to be improved. And also the kernel content of what he has explained that from 20% to 61% was there. If the such kind of genotypes is already available, definitely that can be utilized in the future breeding program. The other speaker, Dr. Hazarika, who is a co-chairman of this session also, is a very learned speaker. And you might have seen his presentation, a lot of genetic diversity, what he has presented. Other time was lesser for him, but he has given a vivid account of the genetic diversity It is special reference to the horticultural crops of the northeastern region, and detailed account he has given about the citrus species, banana diversity, jackfruit diversity, and many wild fruit he has given. Dr. Amarjit Gupta, he has spoken about the genetic improvement of onion program in India, which is also a very important crop, and the kind of variety that the directorate has developed about more than 10 variety. He has given its all characteristics, requirement, and where it can be further promoted. Dr. Anuradha Upadhyay, I know very well to her, and I am very fond of his speaking always. But the time allotted was really very short. I personally apologize for that, and very basic information what she has generated, it can be used in the further breeding program. And now this is the time of the genomic breeding program. Our conventional breeding we cannot replace, but also the genomic breeding program is really very important. And if we go for the genomic data and also the phenotypic data, if we compare both, we can check out certain strategies to start our breeding program. So that is a really very important, very interesting topic where he has a detailed way genomic wide association analysis and linkage methods he has given. But I again thank to him, thank to her. Dr. D. Pandey, he has spoken about the genetic diversity of Aula and Bale. Now these are the upcoming crops. Earlier they were the forest crop, only forest confined crop. But really for the climate resilience point of view, the hardy crops would be given the priority, and some of the work that CISH has done, he has explained here. Dr. R.K. Yadav, okra breeding program. This is a very important crop of India, and very good way he has explained about the present status and future challenges of the breeding program in the okra. And he was the only speaker who has given the credit to the earlier workers, the basic workers who had started the breeding program. Normally, we always highlight, I have done, but really breeding program is not one man strategy. And he has very well spoken, and even the Dr. Harbhajan Singh, Dr. Harbhajan Singh and Dr. Dhankar, all those names he has given. So this way, a very comprehensive presentation was there. Similarly, in the younger speakers, also very comprehensive things were there from different states what accommodated. But anyhow, time was lesser, and also we are running late. So at the outset, I am very much grateful to the ISS and also the all members of the society, and also in particular to Dr. Dwivedi, who has given me opportunity to chair this session. My co-chairman, I am thankful to Dr. Hazarika, who has ably associated with me and noted down all the proceedings. He wanted to also speak, but because of the paucity of the times, I am not allowing. I also excuse. And the reporters had noted down the proceedings. Both reporters, Dr. Alka and Dr. D.S. Mitra. I hope that they have all noted down, and they will make a good proceeding, which we are going to present in the plenary session at the end of this 3-day deliberation. With these words, once again, I am thankful to Dwivedi and his team. Thank you all.
Now, we request our chairman, sir, to present mementos to the matter speaker. The first one is Dr. Sanjay Gupta. Dr. U.K. Dubey, Dr. R.S. Sharma, Dr. Dr.
कोई है फोटो वाला? हाँ, लीजिए.
Dr. U.K. Dubey, Dr. R.S. Sharma,
चले गए होंगे कहीं?
Dr. D.K. Hazarika.
हाँ, लीजिए डॉक्टर. कोई है? इधर ठीक है. आइए. तो जी इधर खड़े होइए, वो फोटो ले. कोई बात नहीं, कोई बात नहीं.
जी तुम्हारा फ्रेंड है.
और
थैंक यू. कहेगा मेरा फोटो.
आइए डॉक्टर. लीजिए. ले जाओ
तो यहाँ पर ना फोटो एडिट कर देंगे।
बहुत बढ़िया।
We request four chairpersons to give momentous
रिपोर्ट आइए तो सभी लोग आए।
आप दीजिए मैडम तो लेंगी। मैडम को यहाँ कहा जा रहा है उधर देखिए कोई दीजिए ऐसे लीजिए।
request Dr. S.K. to give memento to chairman of the committee.
Thank you, sir. Very well conducted despite all this. Thank you. हाँ. This side or this side?
इधर देखिए।
Okay, all the best. Thank you. Yeah, this plenary tea is going on, and then please clear up your cups and bottles because outside we will put outside.
Okay, now we will move.